Hello, Enrique.
What a joy to be here.
Welcome. Good morning.
Thank you very much.
It's an incredible thrill because this is a topic I'm passionate about. Enrique, what is cloning? But explain it to me in a simple way that we can all understand.
Well, cloning is a genetic copy of an animal. A tiny piece of skin is usually taken, or a very tiny grain of rice. And we in the lab, from that piece of skin, we are able to isolate some cells called fibroblasts, which have a nucleus. And on the other hand, an ovum of that species is taken, the nucleus of that ovum is removed, and the nucleus of that fibroblast is inserted. And you have 99.9% of the genetics of that original animal.
Well, look, how's it going?
Look, let me introduce you to David and Marta.
How's it going?
Hi,
Veterinarians.
Marta. How are you doing?
From here, from the project.
All right, David, the first step. What is the first step? And mind you, I want to know something that really well catches my attention. It's done with animals that have passed away and also with those that are alive. It's easier with the ones that are alive.
Indeed. Well, the first step is to perform a skin biopsy, right?
A small piece of meat.
A small piece of meat, basically. And that's it, and then it's automatically washed. And it gets into the means of transportation or maintenance, which are some tubes that carry specific media to preserve the sample.
Some preservatives. The sample is there.
The sample. And then they are sent to the laboratory.
Once the sample is taken, it is crucial for its preservation to take it to the laboratory where we are. From here, Bafosta Samalete, Itaiguera, the process continues.
Hello, Carmen. We are here in the lab where you preserve.
Here, we carry out all the processes related to the preservation of cell lines.
You use nitrogen.
To preserve what cells are, we use nitrogen, which is
At minus 196 degrees.
Temperature is key in everything.
Temperature and the speed, because liquid nitrogen reaches that temperature, the cell reaches that temperature in a very uniform and rapid manner. What that does is that it prevents that ice crystals are formed. Then the cell does not break. The viability of the cell, the survival, well, is very high. And that is the key. How quickly the cell manages to freeze, so to speak.
One of the fundamental steps is the selection of our donors, right? As long as our donor is in optimal conditions, both in age, nutritional, and environmental as well, obviously, it will generate viable eggs and good quality eggs to be used in this process.
How is the egg retrieved from the mare?
Well, the mare's egg cell is extracted through a process that we call OPU, which allows us to aspirate the largest number of eggs possible from a mare, right? Maturation is a fundamental step as it allows our oocyte to have the capacity to host our cell, in this case the cell of the animal we are going to clone, and to achieve a correct cellular reprogramming.
Marta, you're the one in charge of finally taking the sample.
Yes, that's exactly, Yanako. These clone embryos, as they are a bit more delicate, require more ultrasound monitoring throughout the pregnancy. First, the initial check is done to see if it has implanted or not, how many have implanted. Then you look at the heartbeat, and then you continue with more monitoring to ensure everything is going well, placenta, embryo, etc. First weeks of life, because they tend to be born weaker with a slightly weaker immune system and so on. So we are more attentive to them, but later on when they grow up, they develop completely normally, like everyone else.
Enrique!
What happened?
I am fascinated.
Did you like it?
Fascinated. Because we will be able to recover genetics that are already
Of course,
lost. Of course, animals that died and are essential.
exceptional.
Animals that have already died. We have genetic lines of jumping horses, dressage horses, Arabian breed horses. The animals have already died, but the owners froze the genetic lines 20 years ago. So we have that genetic line that we can clone and replicate those animals that are already dead.